Hello from New Zealand, everyone. My name is Anastasija Zaiko, and welcome to my talk. Today I'm going to present the results of our collaborative experimental study where we assessed performance and cost efficiency of eDNA and eRNA capture technique. You all probably know very well that there are multiple challenges and difficulties associated with the collection and recovery of nucleic acid signal from the environmental samples. There are multiple potential sources of variation that are related to variability and type of eDNA and eRNA material available in the water, its uneven distribution, and also different issues associated with the collection and processing of the samples. No surprise, there is a growing demand for optimized sampling techniques and processing protocols for eDNA and eRNA from water samples. In this list of the needed solutions, the upper 3 points are probably of the highest importance and priority for the stakeholders, because these are directly related to the overall efficiency of eDNA, eRNA-based monitoring and surveillance, and also are relevant for the potential future uptake of those techniques. For nucleic acid analysis from water samples, sample filtration is a common and usual step in the workflow, and also often a bottleneck because it requires substantial time effort depending on the water quality and the volumes that you want to process. Usually, we would like to process as much volume as we can to capture meaningful molecular signal, but on the other side, we want to reduce the time of the processing because we don't want to spend hours and hours working with the samples and also compromise the molecular material. There is no general consensus on the protocol, and people are using different pore sizes and different types of membranes, although the report that cellulose-based membranes are usually the best performing for eDNA capture. It is not clear if they are as good for eRNA and also different fractions of environmental nucleic acid material. So this study presents a unique experimental framework of using an easily cultured algal species, Alexandrium pacificum, as a model target to test the performance of different filter types and combining this assessment with the formal efficiency modeling component. To our best knowledge, this has never been applied to eDNA and eRNA studies before. Here, we applied a tiered experimental approach with 2 experimental rounds. In the first round, we aim to assess performance of different membrane types, capturing different fractions of nucleic acids. And those different fractions were produced from the known amount of Alexandrium culture and represented living cellular material or microalgal cell culture itself, a mixture of broken and intact cells, sonicated cells representing shared environmental nucleic acids, and naked DNA and RNA fractions. These 4 fractions of target genetic material were added to the pre-filtered and unfiltered seawater free of Alexandrium. Then 100 milliliters solution of each fraction and water treatment were filtered through different types of filter membranes, and these were cellulose acetate membranes of 5, 1.2, and 0.45 micron pore size, and also positively charged nylon membrane of 1.2 micron pore size. The positively charged nylon membrane was added to test the hypothesis of preferential binding of negatively charged nucleic acids from environmental sample to a positively charged substrate. Finally, the DNA RNA collected on filters were co-extracted, and Alexandrium pacificum ddPCR assay was run on DNA and reverse transcribed RNA samples. The results have shown difference in DNA signal in filtered water treatment, between all three types of starting material: whole cells, sonicated cells, and naked DNA, based on the Kruskal-Wallis test followed by the multiple comparisons. In unfiltered water treatment, DNA yield from naked DNA material was significantly lower from whole and sonicated cells. In terms of the filter comparisons, there was no significant difference between different filter types in the cells and sonicated, full cells and sonicated cells treatments, both in filtered and unfiltered water. However, in the naked DNA, the positively charged nylon membrane showed the highest yield overall. For RNA, in both filtered and unfiltered water treatments, only naked RNA treatment resulted in substantially lower RNA copy numbers compared to cells treatments. There was evident general trend of copy numbers decreasing from intact cells to broken cells and naked nucleic acids for both DNA and RNA and filtered and unfiltered water treatments. And for RNA, again, there was no significant difference between filtered types detected in the whole cell treatment, and somewhat higher yield was reported in the sonicated cells for the PCN filter, as well as in the naked RNA treatment. These findings are also confirmed by the multivariate analysis, ADONIS, and PCA ordination presented on this plot. The overall capture of different fractions did not differ much between different pore size CA filters, but was affected by water used. Naked nucleic acid fractions were more efficiently captured by positively charged membranes. The second experiment was focused on establishing input and output parameters for the efficiency model and was run with two concentrations of pre-sonicated microalgal cells spiked into Alexandrium-free seawater. The spiked water was filtered through the same 4 types of membrane filters, 6 replicates each, while recording time to filter 250 milliliters of water and volume filtered in 20 minutes by continual filtering after the 250 milliliters checkpoint. The DNA and RNA signal was assessed using the same ddPCR assay as in the first round. There was no significant difference in time to filter 250 milliliters and volume filtered over 20 minutes between low and high culture concentrations. However, there were significant differences between filter types, both in volumes and times, high and low concentration treatments. The most apparent difference was between 5CA and 1.2 PCN filters, as you can see. And overall, the PCN filter in this case performed the worst in terms of the timing and volume that could be processed through this membrane. However, in terms of the DNA and RNA signal resulting from the ddPCR analysis, there was statistically significant difference between high and low concentration treatments. The DNA yields were about 2 orders of magnitude higher than those of RNA. And for both DNA and RNA, there was evident general trend of higher copy numbers obtained from the low concentration treatment. However, low concentration treatment DNA copy numbers tend to decrease from 5CA to smaller pore size and PCN filter treatment, while in RNA treatment an opposite trend was observed, both for low and high concentrations, with higher signal detected in the positively charged nylon membrane. For the efficiency assessment, we applied a data envelopment analysis. This is a linear programming technique used for estimating the maximum potential outputs for a given set of inputs. And it estimates efficiency in index from 0.1 to 1, where 1 represents being at the production frontier, This is the solid curved line on the example graph here. And there are two types of efficiency that can be assessed, either input-oriented or output-oriented. And in this case, we pick the output-oriented one to evaluate the efficiency of capturing DNA and RNA by different types of membranes based on the observed volume of sample that passes through within a reasonable time frame. And the overall efficiency analysis indicated that for optimized holistic nucleic acid capture, when the target abundance is unknown, the 5-micron cellulose acetate membrane filter is the best practice choice. The other take-home messages from this work are that coarser pore size membrane captured different fractions of eDNA and eRNA from water, even for targets that are smaller than the pore size, like 5 microns in our case with Alexandrium target. The positively charged membranes show a good potential for enhancing naked nucleic acid yields from water samples, and the water quality and target concentration may substantially affect the nucleic acid signal detection from water. From the efficiency perspective, among 4 tested filter membrane filters, the 5-micron cellulose acetate is the best practice option for optimized capture of waterborne nucleic acids. The presented framework can be easily implemented for optimizing molecular workflow and assessing comparative performance of different sampling and processing tools in the future, not only the membranes, but also other tools and other processing protocols. And thank you for your attention. I'm happy to take your questions now.